In chapter 14, we're going to be covering how genes go from being a gene to a protein, which is our phenotype or the expression that those genes have.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
So remember that RNA is the bridge between DNA and protein synthesis. So RNA can leave the nucleus and DNA is stuck inside the nucleus for eukaryotic cells. And the RNA takes the information that DNA has and makes proteins from it. RNA is pretty similar to DNA, but remember that RNA is called RNA because it has a ribose sugar instead of a deoxyribose sugar. And the base uracil instead of thymine. So in RNA, A will bind with U, and with DNA, A binds with T. RNA is also usually single-stranded, and to be able to get the information from DNA to form proteins, there are two stages. First, RNA has to transcribe the information that DNA has, and then it has to translate it so that proteins can be synthesized. So transcription is the synthesis of RNA using information in DNA. Transcription produces that messenger RNA or mRNA that's able to leave the nucleus. Translation is the synthesis of a polypeptide. So it uses the information in the mRNA to make a polypeptide. And where all of this happens is, where all of the translation happens is in the ribosomes. In bacteria, translation of mRNA can begin before the transcription has actually finished, but we don't see that in eukaryotes. The nuclear envelope has to separate transcription from translation, so transcription happens inside of the nucleus and translation occurs outside. Eukaryotic RNA transcripts are modified through RNA processing to yield the finished messenger mRNA. So here, figure 14.4 in your book shows the difference basically between bacterial cells going through transcription and translation and then eukaryotic cells. So bacterial cells are prokaryotic cells where you do not have a membrane-bound chromosomes or DNA. You don't have membrane-bound genes. So transcription and translation can kind of overlap. Whereas in eukaryotic cells, DNA is bound within the nucleus, and so transcription occurs inside of the nucleus and translation occurs outside on the ribosomes in the ribosomes.
Overview of protein synthesis. Genes are instructions for making proteins. In eukaryotic cells, protein synthesis occurs in 3 steps. First, an RNA molecule is assembled along a DNA template. This step is called transcription. In the second step, called RNA processing, the ends of the RNA transcript are altered. Some portions of the RNA molecule are deleted, and the remaining portions are spliced together to make messenger RNA. The mRNA synthesized then leaves the nucleus and attaches to a ribosome and initiates the process called translation. During translation, the ribosome then reads the RNA message and links amino acids together to form a polypeptide.
Eukaryotic mRNA must be transported out of the nucleus to be translated. The primary transcript is the initial RNA transcript from any gene prior to processing. So central dogma is this concept that cells are governed by a cellular chain of command. And that chain of command is that DNA makes RNA, makes protein. It cannot be reversed. It cannot go the other way. You can't have substitutions. DNA to RNA to protein. So what's interesting about our genetic code is, if you remember, there's only 4 nucleotide bases that make up 20 amino acids. So we only have, starting with DNA, adenine, guanine, cytosine, thymine. And those 4 nucleotides put together will make about 20 amino acids. How many of those nucleotides mixed together make an amino acid? Well, that's going to be 3. So nucleotides form triplets, and those triplets are what make amino acids. So here's kind of a breakdown of how that occurs. The DNA has to be transcribed, and it makes that messenger RNA. Then you take the triplet, the codons from the messenger RNA, and translate that into proteins. During transcription, one of the 2 DNA strands, which is the template strand, provides the template for making that RNA transcript. The template strand is always the same strand for any given gene. And then other places on the chromosome, the opposite strand might function as a template for a different gene. During translation, the messenger RNA triplets, or codons, get read in the 5 to 3' direction, always 5 to 3'. Remember this, because if you are given almost a trick question on an exam or a quiz, and it shows things written in a 3 to 5', you're going to have to read it backwards. So remember, on a strand of DNA or RNA, on a single strand of RNA and on a single strand of DNA, you will see 5' and 3'. And so we read them 5' to 3'. And with DNA, the corresponding strand is going to be opposite, because it's kind of flip-flopped. So each codon specifies the amino acid. So each group of 3 base triplets make an amino acid. So in the mid-1960s, it was discovered that there are about 64 different codons that will translate for the 20 different amino acids. So of those 64, 61 code for amino acids, and 3 of them are dead-end triplets. They end up stopping the signal to end the translation. So why is it, though, we have 61 codons but only 20 amino acids? Well, some of those are redundant. So you may have 3 codons that make 1 amino acid. But it's also not ambiguous, which means that no codon makes more than 1 amino acid. So if I show you the list here of all of the codons that we have for messenger RNA, you can see, I see that there are 4 different codons. So CG, GCG, GCA, GCC, and GCU will all make alanine. But if you look all throughout the rest of this entire thing, you will never find GCU making that codon, making another amino acid. So it is redundant, but it doesn't repeat. So codons must be read in the correct reading frame in order for the specified polypeptide to be produced. They're read one at a time. They do not overlap. And one thing I also want to point out is AUG is our start codon. So wherever AUG, there can be things in front of AUG, like you could have a C, G, lots of codons and lots of nucleotides, that bases that are just nonsense. They are letters that don't mean anything. And you see AUG. Can you now start reading what that polypeptide is? So AUG means you start and then you move on. Also, if AUG is found somewhere within the codon after the start, that becomes MET, or M, that becomes another codon for an amino acid.
The 4 nucleotides of messenger RNA are arranged in 64 three-letter words called codons. The genetic code is a language consisting of these 64 codons. 61 of the 64 codons code for a specific amino acid, while the remaining 3 instruct the ribosome to stop the translation process. Let's see how this table is used to determine the amino acid of a given codon, CAG. First, on the left side of the table, find the first letter of the codon, C. Then on the top of the table, find the second letter of the codon, A. Finally, locate the codon that has a G as its last letter, and you will see that the amino acid coded for by CAG is glutamine. Can you determine which amino acid is coded for by UCA? Serine is correct. Now, can you determine the effect of UAA? The codon UAA signals termination of the elongation of the polypeptide. It's a stop codon.
So what I want you to be able to do is, if you're given a list of, if we start from the beginning, if you're given a DNA template, I want you to be able to take, and I'll say this is your DNA strand. I want you to be able to make the messenger RNA that corresponds with it, go as far back as that, and then I want you to be able to take that messenger RNA strand and translate it into making a protein. You'll have to know that you have to start with AUG, and then you take every 3 codons after that to make another amino acid. You'll also need to know that we have our stop codons, and that once you hit any one of these stop codons, then that means that polypeptide has stopped. So you'll never be given one, I hope, on a quiz or a homework or an exam that is, 25 polypeptides. You'll be given something that's 5 at the most. But you should be able to recognize AUG starts, what happens after that, and know when it should stop. Be able to look at this codon table and tell me what polypeptides are in that protein. So now let's look a little bit closer in how DNA synthesizes RNA. So transcription is the first stage of gene expression. Let's watch a video on how that happens first, and then we'll talk about the stages individually.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that's transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
So remember, an enzyme called RNA polymerase will pry the DNA strands apart and join the complementary RNA nucleotides. RNA polymerase assembles polynucleotides only in the 5' to 3' direction. And then unlike DNA polymerase, RNA polymerase can start the chain without a primer. The DNA sequence where RNA polymerase attaches is called the promoter, in bacteria, and the sequence signaling the end of the transcription is called the terminator. The stretch of DNA that's transcribed is called the transcription unit. There are 3 stages of transcription: initiation, elongation, and termination. Promoters signal the transcriptional start point and usually extend several dozen nucleotide pairs upstream or away from the start point. Transcription factors mediate the binding of RNA polymerase and the initiation of transcription. The completed assembly of the transcription factors and RNA polymerase II bound to a promoter is called the transcription initiation complex. A promoter DNA sequence called the Tata box is crucial for forming the initiation complex in eukaryotes. In elongation, as RNA polymerase moves along the DNA, it untwists the double helix, about 10 to 20 bases at a time. Transcription progresses at a rate of 40 nucleotides per second, and a gene can be transcribed simultaneously in several RNA polymerases. So this could be happening all along a big giant DNA strand at the same time. The mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the messenger RNA can be translated without further modification. But in eukaryotes, RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past that polyadenylation sequence.
RNA processing. In eukaryotic cells, the RNA transcript is modified before translation. This step is called RNA processing. The ends of the RNA, a 5' untranslated region and a 3' untranslated region, do not code for a polypeptide. The actual polypeptide making instructions begin with the start codon and end just before the stop codon. This portion of the RNA is called the coding segment. RNA processing begins with alteration of the ends of the RNA. A 5' cap consisting of a modified guanine nucleotide is added to the 5' end. A poly-A tail made of 30 to 200 adenine nucleotides is added to the 3' end. RNA processing continues with the removal of portions of the coding segment that do not actually code for protein. This process is carried out by small RNA molecules. These small RNA molecules join with other proteins to form spliceosomes. For simplicity, only one spliceosome is shown here. Spliceosomes delete portions of the RNA called introns. They then connect the remaining segments, which are called exons, to form a finished mRNA molecule. The mRNA leaves the nucleus and enters the cytoplasm, where translation of the RNA message into protein occurs.
Genetic information flows from mRNA to protein through a process called translation. A cell translates an mRNA message into a protein with the help of transfer RNA or tRNA. tRNAs move the amino acids to a growing polypeptide in a ribosome. So this is occurring outside of the nucleus. Translation is a complex process in terms of its biochemistry and its mechanics.
Imagine an E. coli bacterium swimming in your large intestine, one of the trillions of bacteria that inhabit your gut. If you eat a lactose-containing snack, such as yogurt or pizza, it will travel through your digestive system, where some of that lactose will likely pass into your large intestine. There, the E. coli will be bathed in and absorb lactose molecules from the surroundings. After it does, the bacterial cell will synthesize an enzyme that breaks down lactose, enabling the bacterium to use this sugar as an energy source. How is the cell able to accomplish this? All cells have DNA that controls the activities within the cell. But DNA never exerts this control directly. That is, DNA itself doesn't transport molecules or break down food. Rather, DNA exerts control by directing the production of proteins via 2 steps: transcription and translation. During the process of transcription, DNA is used as a template to produce a molecule of RNA. At the ribosomes, the molecule of RNA undergoes a second process, translation, to produce a polypeptide, which later forms the final protein. In this case, the DNA gene produces a protein enzyme that breaks down lactose into smaller molecules that the bacterium can use.
The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids.
Each tRNA enables the translation of a particular mRNA codon to a certain amino acid. The tRNA contains an amino acid at one end, and at the other end has a nucleotide triplet that can base pair with the complementary codon on the mRNA. A tRNA molecule consists of a single RNA strand that's about 80 nucleotides long, and it can base pair with themselves. Flattened into one plane, a tRNA molecule looks like a clover leaf. In 3 dimensions, the tRNA is roughly L-shaped, where one end of the L contains the anticodon that base pairs with an mRNA codon. So there you can see that the structure is kind of T-shaped. Accurate translation requires 2 steps. First, a correct match between tRNA and an amino acid done by an enzyme called aminoacetyl tRNA synthetase. Second, a correct match between tRNA anticodon and an mRNA codon. Flexible pairing at the third base of a codon is called wobble, and that allows some tRNAs to bind to more than one codon.
Transfer RNA, or tRNA. The interpreters of the genetic message are molecules of transfer RNA, or tRNA. These small folded RNA molecules act to match specific mRNA codons with specific amino acids. A sequence of 3 bases called an anticodon projects from one end of the tRNA molecule. The amino acid will be attached to the other end. A tRNA with its specific anticodon carries a specific amino acid. There are about 45 different kinds of tRNAs, each with a certain shape and a specific anticodon. Enzymes called aminoacyl tRNA synthetases match each uncharged tRNA with a particular amino acid. The enzyme detaches 2 phosphates from ATP, creating a high-energy bond between the amino acid and AMP. This bond is broken and its energy used to link the amino acid with its particular tRNA, creating an aminoacyl tRNA. Ribosomes are where protein synthesis actually takes place. A ribosome facilitates the pairing of each mRNA codon with a particular tRNA anticodon, and then links amino acids in the sequence spelled out by the genetic message. A ribosome consists of a large subunit and a small subunit, made of protein and ribosomal RNA, or rRNA. The subunits actually join only when the ribosome is associated with an mRNA molecule. 3 sites on the ribosome hold tRNAs during the protein-making process. The P site holds the tRNA with a growing polypeptide. The A site holds the tRNA with the next amino acid to be added. Used tRNAs exit via the E site. This is a more realistic illustration of a ribosome.
Ribosomes facilitate specific coupling of tRNA anticodons with mRNA codons during protein synthesis. The large and small ribosomal are made of proteins and ribosomal RNAs. In bacteria and eukaryotic ribosomes, the large and small subunits join to form a ribosome only when attached to an mRNA molecule. A ribosome has a binding site for mRNA and 3 binding sites for tRNAs. There's a P site, an A site, and an E site. The P site holds the tRNA and carries the growing polypeptide chain. The A site holds the tRNA and carries the next amino acid to be added to the chain. And the E site is the exit, where discharged tRNAs leave the ribosome. So P site carries the polypeptide chain, A site, amino acids added, E site, exit site. The 3 stages of translation, just like in transcription, are initiation, elongation, and termination. And all 3 stages require protein factors that aid in the translation process. Of course, the energy is provided by hydrolysis of not ATP, but of GTP.
We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with a large ribosomal subunit to form an initiation complex. The initiator tRNA sits in the P site on the ribosome. Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule. Part of the ribosome catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated. Three special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Mutations are changes in the genetic material of a cell, and so we can have point mutations, which are chemical changes in just one nucleotide pair of a gene. The change of a single nucleotide in a DNA template strand can lead to the production of abnormal protein. It just takes one thing to be wrong for that whole protein to not be correct. If a point mutation occurs in a gamete, it can also be transmitted to the offspring. And this is something that we see with sickle cell anemia. Sickle cell disease is a really debilitating disease, in the United States. And you might ask yourself, why is this mutant? It's a mutation where the red blood cells are not nice and kind of donut-shaped, pillow-shaped, like normal blood cells should be. Instead, they're sickle-shaped or caved in, kind of almost dehydrated looking. And this is a very painful disease for those individuals that have it. And so you might wonder, why on earth is that mutation still in the population? And one reason that it is because in places like Africa, where malaria is very prevalent, having sickle cell anemia is actually a benefit, because the parasite cannot get into the blood when it's shaped like a sickle. But sickle cell anemia is a mutation that has proven to actually be beneficial in places where malaria is prevalent. However, in the United States, where we don't have malaria, it's kind of a lingering mutation that someone has brought over from areas where it is a benefit. And here it's not so much of a great thing to have. Small-scale mutations within a gene can be divided into two general categories. You can either have a single nucleotide pair substitution, or you can have insertions or deletions. With the substitutions, basically all you're doing is just swapping something out. You're replacing one nucleotide and its partner with another pair of nucleotides. Some of these can be silent mutations, which means that they just have no effect at all on the amino acid. And it could be because of redundancy in the genetic code, but basically that paired substitution doesn't have any negative or positive effect. Missense mutations will change one amino acid into another, and most substitution mutations are missense mutations. Nonsense mutations will change an amino acid codon into the stop codon, and so it truncates the protein from the way it should normally be, and this will nearly always lead to a non-functional protein. Insertions and deletions are additions or losses of nucleotide pairs in a gene. These mutations will normally have a disastrous effect, more so than the substitutions do, and they may alter the reading frame of a genetic message, and this produces a frame shift mutation. Spontaneous mutations can occur during DNA replication, recombination, or repair. Mutagens are physical or chemical agents that can cause mutations, and researchers have developed a lot of methods to test the mutagenic activity of chemicals. So many chemicals that we consider carcinogens, which are cancer-causing chemicals, are mutagenic.
Types of small-scale mutations that affect mRNA sequence. This figure shows how different types of small mutations in the DNA sequence of a gene affect the mRNA sequence and, in many cases, the resulting protein. Let's look at the wild type or most common DNA sequence, shown in the two blue strands here. Notice that each nucleotide, identified by the base it carries, is shown across from its complementary nucleotide on the other anti-parallel strand. The red single-stranded mRNA is transcribed from the DNA template strand, which is the top blue strand. You can see that the mRNA sequence is a copy of the other strand of DNA, the bottom strand, except that it has U's instead of T's. The purple strand shows the sequence of amino acids in the encoded protein. Each triplet codon of nucleotides in the mRNA is translated by a ribosome so that a specific amino acid is added to the polypeptide chain. For example, the codon AAG specifies the amino acid lysine, abbreviated LYS. When a mutation occurs in the DNA sequence, this also changes the mRNA sequence, which may or may not change the amino acid sequence. Let's look more closely at the range of effects of small-scale mutations. First, let's look at nucleotide pair substitutions. They're also called point mutations because the sequence is changed at only one nucleotide. The first example shows that a G has been changed to an A in the top DNA strand. After DNA replication, its complementary nucleotide partner has now become a T instead of a C. When the gene is transcribed, there will be a U on the mRNA in that location, complementary to the A, instead of the C in the mRNA transcribed from the wild-type gene sequence. But what about the amino acid sequence? Will it be changed? To see what the new codon codes for, let's circle each triplet codon from the start to identify which three bases make up the mutant codon. The fourth codon is now GGU instead of the GGC in the wild-type sequence. Let's look up GGU in the mRNA codon table. The first base is G, the second base is G, and the third is U, so we end up here on the table. GGU codes for gly, which is the amino acid glycine. Note that the GGC in the wild-type mRNA also codes for glycine. So the sequence resulting from this mutation isn't any different from the wild-type amino acid sequence. In both cases, the codon codes for glycine, and the resulting protein is exactly the same. We call this kind of genetic change a silent mutation because it has no observable effect on the phenotype of the organism. Mutations can be silent because more than one codon codes for each amino acid. In other words, because the genetic code is redundant. The next nucleotide pair substitution example is not a silent mutation. The wild-type C in the fourth codon was mutated to a T, which changes the G in the wild-type mRNA to an A in the mutant mRNA. The mutant fourth mRNA codon is AGC. What amino acid does the mutant codon AGC code for? AGC codes for ser, the amino acid serine. The mutation leads to a change in the amino acid sequence of the protein. In the wild-type protein, the fourth amino acid is a gly, but in the mutant, it's a ser. We call this a missense mutation because it codes for a different amino acid, which might have different chemical properties that affect protein function. The last kind of nucleotide pair substitution is more severe. In the second mRNA codon, the A of the wild type is a U in the mutant. When you look it up on the codon table, you find that the lyse of the wild type has been replaced by a stop codon in the mutant. The protein synthesis machinery will stop adding amino acids as soon as it gets to the stop codon. A mutation that causes a stop codon is called a nonsense mutation, and most lead to shortened non-functional proteins. Those are the three kinds of nucleotide pair substitutions, or point mutations. Now let's examine insertion and deletion mutations. The first example shows a single nucleotide pair insertion. There are 16 nucleotide pairs in this strand compared to 15 in the wild type. An extra A and its partner T have been inserted into the sequence. This results in an extra U being added to the mRNA. Now let's circle the codons in the mutant sequence to see what amino acids will be in the polypeptide sequence. Notice that the extra U causes all of the codons after it to shift over one space, so that all of them are different from the wild type. We call this a frame shift mutation because it shifts the 3-nucleotide reading frame the ribosome uses when translating the mRNA. The insertion is early in the sequence, and now the second codon is a stop codon. So the insertion causes both a frame shift and the resulting nonsense mutation. The next example shows what can happen when a nucleotide pair is deleted from the DNA. The A is deleted from the DNA along with its partner T, so the mRNA lacks a U, and the codon reading frame is shifted after the place the U should have been. This time the frame shift doesn't code for an early stop, but instead causes the sequence of amino acids after the deletion to be different from the wild type. This is both a frame shift and a string of missense mutations, and eventually in the sequence there's likely to be a random stop codon. What happens if more than one nucleotide pair is deleted at a time? If the number of nucleotide pairs is not a multiple of 3, it will cause a frame shift like the one in the previous example. But if the deletion is a multiple of 3 and doesn't affect the reading frame, like in this example, then it will cause a whole codon or codons to be lost. The resulting protein will have fewer amino acids. In this example, one codon, AAG, is missing, and so the amino acid lyse is also missing from the protein. But amino acids after the missing lyse are the same as the wild-type protein. Likewise, if insertions of multiples of 3 nucleotides results in additional codons, then the resulting protein will have extra amino acids. However, if the insertion coded for a stop, it would cause a truncated protein. So small-scale mutations can be either substitutions, insertions, or deletions, all resulting in altered mRNAs. Depending on the mutation, this may alter one or more amino acids or even shorten the length of the protein, and can therefore have widely varying effects on protein function.